Welcome to Humanley, the podcast providing allied health and integrative medicine practitioners with the most up-to-date, evidence-based and clinically relevant information. This podcast is a melting pot of ideas on health and well-being and does not replace the advice of your primary healthcare professional. Here's your host, Daniel Reuters.
Hello, everybody, and welcome to the Humanley podcast. My name is Daniel Reuters, and today I'm joined by Professor Gerald Pollack. Professor Pollack, thank you for being here. It's so wonderful to have you on the podcast.
Well, thank you. I'm delighted to be with you, Daniel. Very happy.
Professor Pollack, you have been researching structured water for over 20 years, and you're currently a professor at the University of Washington in Seattle. Can you start off by explaining just exactly what structured water is and what actually got you interested in studying structured water?
Sure. Happy to do that. Well, the term structured water, it's an old term. We really don't use it so much these days, but the idea is, because everything has structure, so it's slightly ambiguous. But I think the intent originally was to distinguish a kind of water from ordinary water that's in a glass. See, ordinary liquid water, the current understanding, which by the way is incomplete about liquid water, is that liquid water consists of individual molecules that are randomly disposed relative to one another, and they're bouncing around at a fierce number of times per second, perhaps per femtosecond, and that's the current concept of liquid water. So structured water actually has structure to it. It's not, the molecules are not bouncing around that way. They're not randomly oriented. They're actually ordered in some way, and the way they're ordered has not been so clear. Some ideas have come into being, and we've extended those ideas or modified those ideas, and I will tell you about that if you ask me in a bit. But that's the difference between so-called structured water, in which the molecules are. You might say some people have advocated the idea that the molecules are kind of like standing at attention, like soldiers standing at attention, organized in some way. Unlike liquid water, where they're at ease, moving around, so that's essentially the difference. And the reason I got interested is, you might say, is a quirk of some sort. We had been studying the mechanism of muscle contraction, and the mechanism that's in the textbooks today is not so different from was in the textbooks 30 or 40 years ago. And there's a problem that we were working on, and the problem is that, unfortunately, that the evidence doesn't fit the theory, or a theory doesn't fit the evidence. And we spent quite a few years doing experiments, some of them, I think, fairly sophisticated. We got known for our advanced apparatus that could measure phenomena that other people couldn't measure, and almost nothing fit the predictions of the theory. It was a bit of a problem for us because if you want to put it in a certain way, our competitor, if you will, it was a famous scientist, Sir Andrew Huxley, who won a Nobel Prize. His Nobel Prize was given for his work on currents and membranes. And then he liked microscopes and he liked fiddling around with muscles, so he got into that field. And he is the person who set out the theory. I wrote a book on muscle contraction in 1990, and I laid out the reasons why it's called Muscles and Molecules, the reasons why the theory didn't work and what might actually work better. And I still think that there's a measure of truth to what I put forth. But somehow we knew that water was centrally involved. Now, water plays no role whatsoever in the current understanding of muscle contraction. It's the proteins, myosin and actin, that somehow interact with one another. But the odd thing that we came to realize is that the muscle is mostly water. It's not mostly proteins. If you think of the volume of a muscle, like the rest of the body, pretty much, it's roughly 2/3 water. And that 2/3 is 2/3 by volume, right? If you were to, and because the water molecules are so small, in order to feel that 2/3 volume, you've got to pack in a lot of water molecules. And if you do the count, if you could take all the molecules that are sitting inside a muscle and line them up one by one and start counting, more than 99 out of 100 would be water molecules, because the proteins are very large. The water molecules are small, so you need many of them. And we knew that water had somehow to be involved. And then the invitation to present at a conference, and this conference was held in Hungary, and it was to commemorate the work of a famous Hungarian biophysicist who had passed recently, and he had two interests, and one interest was muscle, and the other interest was water. So I was invited to represent the muscle contingent, although the water was interesting for me as well. And I presented our work on muscle. And I think, if I recall, it was reasonably well received. But what intrigued me there was the presentations on water, because the most prominent person who was there was a guy named Gilbert Ling. And I had known of Gilbert Ling because I was a graduate student at the University of Pennsylvania, and he was in Philadelphia at the same time. I even saw him once at a conference, and someone pointed out to me, hey, the guy sitting 2 tables away, he's that crazy guy, Gilbert Ling, who has an idea that inside the cell, the water is not like liquid water. All the molecules are lined up in some way. So he used the term structured. So I sat and I listened to Gilbert, and I also listened to perhaps a dozen other people who had come and who had evidence to support this point of view by Gilbert Ling. And of course, I made friends with all of those people, and I was seriously impressed. And I came home excited. I came home from Hungary wanting to share this new understanding or new information with the students and postdocs in my laboratory. And I gave them one of Gilbert's books. By that time, he'd written 4 or 5. And of course, it was extremely controversial, no doubt about it. And every one of them came back to me and said, this is really interesting stuff. And if this guy is right, it changes all of biology. I mean, completely. And so that reinforced the notion that this was something revolutionary, but revolutionary in a positive sense. That Gilbert has his finger on truth, or close to it. And so I'm easily distracted from what we had been doing, and I quickly saw that this has relevance not only for muscles, which we were working on, but for all of biology, and maybe even beyond. So first thing I did was I decided I'm going to write a book. Now, Gilbert had already written a whole series of books on the subject. And I should point out, by the way, that Gilbert was chosen among all students in China. There were 3 who came after World War II to study in the U.S. There was a chemist, a physicist, and a biologist, and Gilbert was chosen as the biologist. So this was a highly select group of people, not the ordinary run-of-the-mill. These were the top people in China, and invited to the U.S. And they all thought Gilbert was the smartest of the three, or so the rumors were. And one of the guys, his name is Yang, he went on to win a Nobel Prize in physics, became famous. I think he became famous not only because of his Nobel Prize, but as the story goes, he married his translator, and he was something like 90 years old, and she was 30 or something. So that got a lot of notoriety. But anyway, I just wanted to digress for a moment to tell you that Gilbert, who passed less than a year ago, almost reaching 100, was a really special guy. Not necessarily the easiest to deal with, but brilliant, totally brilliant, and certainly should have won a Nobel Prize. So one of the problems, though, with Gilbert Ling is that his writing is not the best, not so clear. I can imagine Gilbert sitting, although I'm not sure, sitting at his typewriter or word processor and batting it out and sending it to his publisher. You see, and it doesn't work usually when you do that. The number of revisions. I was just reviewing a book proposal by someone who's writing about about science. And he talks about that in his particular field, it's not unusual to rewrite something 50 times.
Wow.
Yeah, I couldn't believe what he was saying, but it's not surprising because one of my students, we're working on a paper, and I think we're about version number 30 right now. And it's driving me nuts, but we're getting there. So anyway, excuse me. His works, I wouldn't say are impenetrable because you can penetrate, but it sure takes a lot of work to get through and understand. And if you're a physical chemist, you have a better chance of grasping some details. And if you're not, then it's not so easy. So I decided to write a book describing Gilbert Ling's work. And it's still on Amazon, actually, surprisingly selling well. And that was my 2001 book, not the latest book, 2001, called Cells, Gels, and the Engines of Life. And I described how Gilbert Ling's ideas on structured water inside the cell. But I went on, this was maybe a mistake because Gilbert didn't appreciate this so much. I went on a step further to describe how this idea of structured water was critically important for every cell in the body, not just muscles, but nerve cells, secretory cells, what have you. And the central point in that book was to adduce evidence to demonstrate that the conversion, the transition, we say phase transition between structured water and ordinary liquid water is a trigger for so many cellular events. For example, in muscle contraction, the relaxed muscle contains this kind of structured or ordered water. But when the muscle is ready to contract, what happens is that there's a phase transition, that the water undergoes this transition from the structured water, which I'll tell you more about, to ordinary water. And then that enables the proteins to undergo conformational changes or folding, if you will, to manifest the contraction. And then when the contraction is over, the water and the proteins return to their initial state and back again to the so-called resting state or relaxed state. And the same thing is true in other kinds of cells, like nerve cells transmitting information. There's a transition in the water inside the nerve cell, which allows the proteins to undergo a conformational change, and the information is then transmitted along the nerve cell. And the same with secretory cells. Phase transition, water changes, structure changes, and the products are ejected from the secretory cells in something like an explosion. So that was my entree into the field, and the last step in the story is that, of course, having written this book, and the book was received with mixed mixed reviews. Anybody can read on Amazon. Some people said, some reviewers said, this is pure nonsense. It's just an extension of Gilbert Ling, and everybody knows that Gilbert Ling is a crazy guy or something like that. So it's just a nonsense. And I remember another review saying, "Oh yeah, Pollack. I know this guy. He always complains that the establishment is causing him grief." And I think at that stage in my life, I was never complaining about that. And another guy from Harvard, head of a big group, writes, "This was a real pleasure. I should have hung it up on the wall." He says, "This is a 304-page preface to the future of cell biology." Of course, I like that. So anyway, we decided that we have to start doing experiments to get into this, and within 6 months or so after publication of that book, I came into contact with a Japanese guy I never met before, and he told me something that was a really critical clue as to how to find a preparation that we could use to study, and we started, and that's how it all began. So instead of going on and on and on, I'll stop because I'll wait for you to ask questions.
That was fascinating. Thank you for sharing that.
Pleasure.
That brief history. For our listeners who've never been exposed to this topic before, they may not know that there actually is a fourth structure of water or fourth phase of water. So are you able to explain a little bit more about what those four phases of water are and why structured water, or easy water, I think some people call it, why it's so important or more important than the other three phases?
Sure. Well, I'm not sure about more important, but this is more than just a laboratory curiosity. It's actually this fourth phase, which I'll tell you about, is what fills our body. It fills our cells, and as I mentioned before, there's a lot of it. And so, if you really want to deduce the working mechanisms inside the body, you obviously need to know what's inside the cells, and this fourth phase is essentially what fills the cell. So what's the fourth phase? Well, so the three phases that we learn about are solid, liquid, and vapor. And I think I don't have to explain what they are. They're, the solid is ice, and the liquid water we drink, and the vapor is what rises to form clouds. And there had been talk about a different kind of water, a different phase of water for more than 100 years. And the reason for it, one reason for this is that if you start with if you presume that water has only these three phases, it's really difficult to explain so many phenomena. At the time of the beginning of the 20th century, there was a substantial number of these, and by now, if you look on the web and try to, there's a website that contains, there are something like 60 or 65 anomalies of water, and this is based on the idea of something that doesn't fit the concept of three phases. So the idea is that, well, perhaps there's a fourth phase of water, and people have talked about this, and even I mentioned Gilbert Ling a moment ago, but even Albert St. Georgi, who is often considered to be the father of modern biochemistry, he was talking about, he didn't say a fourth phase, but essentially he was talking about structured or ordered water, which is essentially the same. And one of the many famous quotes from St. Georgi, who, by the way, discovered vitamin C and studied muscles and water and many things, he said, life is water dancing to the tune of solids. It's one of those, ones you don't forget. So there has been a lot of thinking along these lines, although the kind of thinking has been considered radical thinking, because it's simpler to think about the cell as bounded by a membrane and stuffed with ordinary liquid water. However, anybody can demonstrate that that's not correct. And the simplest way is to cut yourself. And if you cut yourself, if your cells are filled with liquid water, all that water would pour out, and you'd lose your water. You see, but that doesn't happen. The water is more inside the cell, is more gel-like, and that's what this kind of ordered or structured water is. So let me go a step more and explain to you what we found and why that's somewhat different from what Gilbert Ling had been espousing, and why it's important. Okay, so what we found, this doesn't have to be inside the cell, it could be anywhere, is wherever you have water. We found that when the water falls adjacent to some material, if that material is hydrophilic or water-loving, not every water-loving surface, not every hydrophilic, but many, if not most of them, the water undergoes a change. So you start with ordinary water molecules, and those water molecules that are impacting this material surface undergo a radical transition. They go from individual water molecules to a sheet. And the sheet is a hexagonal or honeycomb sheet that sticks on to the surface. And then another layer forms subsequent to the first layer, and then a third layer, and so on. And these layers keep building. And they can build out to, under the right conditions, we've seen them building out to up to like 1 million layers or something like this. This is a very impressive kind of buildup. It's not trivial by any means. Inside the cell, the buildup is obviously not that much, because the space between solids inside the cell is only maybe on average a distance equivalent to like 7 water molecules lined up. So there's not much space. So this kind of water, the reason we call it fourth phase, but we also call it exclusion zone. And in fact, it was a guy from Brisbane, a friend of mine, who suggested that name. Odd coincidence. He said, you got to give it a name, because, you're seeing this all the time and nobody else has seen it. Parenthetically wrong. It was actually observed 30 years before we observed it by a group of physiologists looking at the cornea of the eye. So we called it exclusion zone because this kind of water is like a crystal. It's organized, and crystals exclude solids. So, for example, when ice is forming, the forming ice excludes the solutes. Otherwise, you wouldn't get a pure crystal. And ice is generally a pure crystal. So you can see, for example, a glacial moraine ahead of the glacier of ice. And so because this water we found experimentally excludes, first we found it excludes particles, then various molecules and such. It's a practically universal excluder. So John Watterson was the guy from Brisbane, physical chemist, and he said, you should call it exclusion zone because it excludes. And then the water that's inside that zone is exclusion zone water. And he said it has a real advantage, and the advantage is that it's exclusion zone EZ. It's easy to remember, but that works in the US. I'm not sure about Australia, but in most places it's EZ, not EZ. So it doesn't work. Yeah, it doesn't work quite as well. Anyway, the name stuck, and in a way it's an unfortunate name because it just reflects one of many, many properties of this kind of water, the fact that it excludes. So one more thing that I need to mention, because it's critically important, is that this water is not neutral anymore. It's actually typically negatively charged. And so as these sheets build, they build a zone of negative charge, and beyond the region where they've built up is ordinary water, and that ordinary water is filled with the complementary positive charge. And the reason for that is all of this is built from water molecules. And the idea is that the water molecules are then broken into two components, H plus and OH minus. And it's those OH minuses that gather together to form this exclusion zone or fourth phase, while the positive component, the protons, are sitting outside in the ordinary water. So you have a battery, essentially, where the exclusion zone is negatively charged and the region beyond is positively charged. And we've demonstrated that you can actually get electricity from this battery, one of the interesting developments. So finally, a question is, finally in what I'm describing, in order to build this ordered zone, you're going from disorder, from chaos to order. In order to do that, you have to supply energy. You're also charging battery at the same time. In order to do that, you need energy. Your smartphone won't work tomorrow if you don't plug it in tonight. So you need energy for both of those phenomena, both of those aspects. And I tell you, we couldn't figure out where that energy was coming from. We spent several years of head scratching. And finally, it was a young undergraduate student who led us to the right answer. And he was doing something that he wasn't supposed to do, which is great. We encourage that. It's sort of like living in Australia. We encourage. So he was studying the exclusion zone in a little experimental chamber, and sitting next to him was a lamp, a gooseneck lamp, and he decided to shine the lamp onto the preparation, and then he called me to take a look, and what I could see was that where the light was incident on the chamber, the exclusion zone was hugely expanded. So I thought, wow, this is pretty interesting. It looks like light, photons, are really the source of energy for all this. And we went on to do, of course, real experiments, and we tried to find out which wavelengths of light were important in building this fourth phase of water. And we found that it was not the visible wavelengths, starting from violet all the way to longer wavelengths of red. It was beyond red. It was infrared. Infrared wavelengths were powerfully effective in building this. We could take an LED, a light emitting diode, very, very weak, barely see the light, and shine it on the chamber, and over a period of an hour, it would increase by 10 times. So it's really powerful, and you take it away, and it recedes back to the original. So it's very powerful. So to summarize, because I've gone through a lot of stuff, the evidence that we have is that this fourth phase consists of sheets, hexagonal sheets that build on one another. They have negative charge, and the region beyond has positive charge, making a battery. And all of this is built by mostly infrared light that's incident on the preparation. And finally, this is what fills our cells. Okay, so let me stop there and let you put in a few words edgewise.
Wow, that's amazing! Amazing what you've found there, and I've got lots of questions now swimming around in my mind. So you're saying that this exclusion zone water, the way you're describing it, I was immediately thinking that it's possibly taking the place of the sodium-potassium pump of cells, or am I thinking of two completely different ways in which things move in and out of cells now?
Well, your question is actually a little bit complicated, although the answer is maybe simple. So let me address the sodium-potassium pump, and let me preface it by saying that I believe there is no such pump, and it's not my original idea. This is an idea coming from Gilbert Ling. So, and what he found is, if you have a pump in the membrane, you need energy to power the pump, like any pump. Always needs energy. And so Gilbert doubted that the existence of this pump for various reasons, and he did a critical experiment, which has never been questioned by anybody. People essentially don't refer to it, but the experiment is pretty clear. He poisoned the cell with a cocktail of poisons, so that the cell was really poisoned. And then he looked. So in doing that, he said, well, if I poison the cell, I'm depleting the cell of the energy that's required to power the pump. And so all of the effects of pumping should vanish, but they didn't. He did some computations, and the computations show that for the sodium pump, in the most generous of presumptions, generous to the opponents, the cell would need something like 30 times the amount of energy that that could be supplied in this condition. So it clearly couldn't work unless there was something wrong with this experiment. And as I said, nobody has challenged it. Well, since the advent of the so-called sodium pump or sodium-potassium exchanger, there have been discovered, I think, something like 2,000 additional pumps. I may have that number wrong. It may be 1,500. I asked a student of mine who went into that field, and he listed the number, and it was something like that. Huge number. They all require energy. And so if the cell is, the computation was that the cell was using, the cell that was not poisoned was using something like 1/3 of its energy to power the sodium pump. Now you've got 2,000 pumps, and where's the energy going to come from? The cell doesn't have enough energy to do anything, or even to power those pumps. So Gilbert argued that this is wrong, and I agree with his point of view. So he's challenged the establishment, and it's also difficult to find them. You look at electron micrographs of the cell and the cell boundary, the cell membrane, and you ought to be able to see those proteins because you can see other proteins, but yet the membrane in the images looks like a railroad track, with two rails, or actually three rails, and it's not interrupted by these structures. And also a final argument, maybe it's not the final one, but Gilbert argues there's no real estate. There's so many pumps, and there's no place in the membrane to put all those pumps. And also, just one more. Apparently, the conclusion that's been reached is that even the chemicals, the drugs that are produced by the pharmaceutical companies, which the cell has never seen before, the conclusion is that there are pumps for those too. And how on earth the cell could muster a pump for a chemical that it's never seen before is a challenge. So there are many reasons why the pumping doesn't really occur. And so the second part of your question is, well, what about EZ or fourth phase water? And you're right on. And so the sodium potassium exchanger is said to be responsible for the negative charge of the inside of the cell. So you take a pair of electrodes, you stick one micro electrode, parenthesis, originated by Gilbert Ling, same guy, should have won a Nobel Prize, because it's come widely into use. So one microelectrode is inside the cell, another one's outside the cell. And you can measure a potential difference. Typically, it will be somewhere between 50 and 100 millivolts negative inside the cell. So why is it negative inside the cell? Well, the textbook explanation, which has to do with sodium-potassium exchange, is that the cell is exchanging ions in such a way that more negative ones remain inside the cell than positive ones, and you wind up with a negative electrical potential inside the cell. But that's difficult to square with evidence. The first is if if the system of pumps is not working, then there's something that is not right. And the second is if you cut off part of the cell, in other words, break the membrane, break the cell, you still can have the effects that you would ordinarily attribute to the membrane pump, even though you've opened the cell to the outside. So the cell should have zero electrical potential, but it doesn't. It maintains its electrical potential for a long time. So what's going on? Well, I think what's going on is really simple. It's not complicated like the pumps. What it is that the water inside the cell is easy water, which has negative charge. So, of course, if you were to stick an electrode inside the cell, you'd measure a negative electrical potential, period. I think that's the correct explanation for what's going on. I discussed this a little bit more in my later book, cells, gels, the fourth phase of water, which has become really popular. I feel so gratified, but there's more discussion of that in that book, along with so many other things. So I think I should stop there with that question, because otherwise I could go on forever.
It just makes so much sense the way you're explaining it. It really does. It just resonates with me, what you're saying, and
well, thank you.
It's shaking my foundations of my understanding of human biology and how I've been taught that cells work. So that's amazing. I love being challenged. I love having these amazing new thoughts being presented.
Well, thank you very much. Really appreciate your comment, Daniel.
No, I thank you for providing the information. It's wonderful. So this brings me then to our next question. If we're getting energy from light or photons of light to power the cells, what's a mitochondria for? Obviously, our understanding of that is not correct either.
Well, you see, not exactly. I think that's accurate. I myself have not studied mitochondria in great detail, but I can say a few things about the mitochondria. If you look at the structure of a mitochondrion, it's full of membranes, internal membranes, and those membranes are interfacing with water. And so I can easily imagine that the water inside the mitochondrion is easy water, because the situation is exactly right. Now, easy water is full of negative charge. And so if this negative charge is then supplied to the cell, we have evidence that negative charge builds easy water. So you stick an electrode in a bath of water, and if you pass current through, the negative electrode builds easy water right around it. The charge pours into the water, and you get a buildup of easy water. Now, the cell, of course, the cell needs the easy water. It needs a full complement of this water to function properly. So it might be that the mitochondrion is a factory that produces negative charge. The negative charge leaks out into the rest of the cell, and that negative charge builds easy water. So the mitochondria are there to ensure that the easy water is there. And remember, this water has energy, potential energy, because the cell is filled with this negative charge, and all those negative charges want to get as far away from one another as possible. They repel, right? And that's energy, that's potential energy that could then get delivered. So I do think that the understanding of energy inside the cell is not exactly as the textbooks have it. And the evidence we have is that electrical energy in this form that I just mentioned to you is critically important for driving biological processes. Whether that's the main source of energy, I'm not so sure, but I don't know. Nobody knows, but it's certainly one component of the energy. So so this is I think maybe really significant. The energy provided by the mitochondria could be electrical in nature.
So with easy water, is this a substance that we can obviously produce endogenously or within ourselves, but do we need to get structured water from our external environment as well? Or is it sufficient just to drink any type of water and then our body will do the rest?
Well, I think it's true that the body will do the rest in a healthy person. You need nothing more, and you've got energy coming from sunlight. Sunlight, more than 50% of the energy from the sun is, as you well know in Australia, is infrared heat, you might say. In infrared energy, and that's the energy that is necessary for building this kind of water. So yes, in theory, there's plenty of energy from outside coming in. In addition to that, metabolism is generating heat, and the heat that is generated is essentially, not exactly, but essentially equivalent to infrared energy. So you have in your body, you've got external sources from the sun, and internal sources from metabolism. And by the way, the external sources is not just directly from the sun. Everything around you in your environment is generating infrared energy. And the evidence for that is in your room or laboratory or bedroom or whatever, turn off all the lights and you see nothing. Your smartphone won't be able to get any image, nor will your retina get any image. But if you whip out an infrared camera, that is a camera whose sensor is sensitive to infrared, you get a beautiful image of everything that's in your room or outside. And that's why it's used in the military in the dark to see, the enemy, because you can pick up all of that. So the energy is there. It's from the sun. It's from everything in your environment, and also from inside your body. So if you drink the water, the energy is coming in. It builds easy water. That should be all you need. However, some cells that are dysfunctional or even pathological, one of the causes of pathology, and I think it could be a major one, is what people refer to as dehydration, and that is not enough water inside the cell, or not enough EZ water inside the cell. And the evidence for that is people 50 years ago, I was doing this at the beginning of my career, poking cells with electrodes to look at the negative electrical potential. And a typical healthy cell will be minus 70 or minus 80 millivolts or something like this, negative. But pathological cells are much less negative. So studies both on cancer cells, those are the ones I can remember, and pathological kidney cells, they both show electrical potentials minus 10 or minus 15 millivolts. And according to what I was trying to relay to you earlier, this electrical potential arises from the water, the negatively charged water. And if you have a potential of minus 10 instead of minus 70, it means there's less EZ water inside the cell. And so a possible origin, if you will, of the pathology is not enough EZ water. So you want to restore the EZ water. And of course, the body can do what it can itself to restore it, but you can help. And there are multiple ways of helping. I think I've been simple ways of helping. I made a list, and I think it's published, some very simple expedients that anybody could employ to improve their health. And these do improve health. And if you ask me, I can go down the list, but that wasn't your question. Your question is, can you produce it endogenously from the raw material of water, or must you get it elsewhere? And you can get it elsewhere. I'll just give you one example of, as I said,
Yeah, I'd love to hear some examples, Dr.
Well,
Pollack, please.
okay. So I run down the examples briefly. One of them is drinking water. Obviously, it's the raw material. You need it. The second one is juicing. So, by juicing, I'm talking about the expedient of going out in your garden and taking some leaves from some plants, some freshly grown leaves, and then mashing them in a machine or by hand and squeezing out the juice and drinking the juice. So what are you drinking when you drink the juice? You're drinking the water from inside the plant cell, and that's just full of easy water. These are healthy leaves that have just come into being, plenty of sunshine, and so by drinking this water and trying to make it palatable by adding a little bit of flavor of some sort, you're basically directly providing easy water to your body, and so you can offer this easy water to the cells that need it, and the cells will take it up and build easy water, and you will feel better. And the evidence for that, I haven't seen published evidence very much, but the health providers that I've spoken to, a fair number, what they report is the patient has a problem, and the therapy that they suggest, one of the therapies or the major one, and sometimes only that, is to drink the juice of the plants. And the reports I get are that the people come back, they return after a few months, and whatever was ailing them is not ailing them nearly as much or at all as when they came in. And byproduct is also weight loss. So you've got both. And all of this is anecdotal, but it makes sense to me because you're drinking easy water and you're replenishing the easy water that's missing in your body, and therefore you're promoting function where before there was dysfunction. So that's number two. Number three is herbs of various sort, natural substances. So all of this dates back to Ayurvedic times or ancient China, when the people were maybe smarter and they knew how to heal themselves. And in this tradition, take the Ayurvedic tradition, for example, what has lingered and what has come out of that tradition is a couple of substances that lead the way. One of them is turmeric, and another one is basil, holy basil, and so those are known to have positive effects on health. And beyond that, they're known that it's not just one specific aspect of health, like reversing a kidney pathology or something. It's all over the body. Practically whatever ails you. Take turmeric. It's possible. There are two hypotheses that you can, I think at least two, that you could put forth. One is that the turmeric is really special, and it can impact the receptors of your kidney cells, your heart cells, your nerve cells, or whatever. Or another hypothesis, instead of having multiple mechanisms, there's one mechanism, and that is it impacts the water, which is all over, which then can impact all of those organs. And so naturally, we thought the second one is a simpler hypothesis, and based on the principle of Occam's razor, we're drawn towards simplicity. And so we began testing. So we tested turmeric. We set up a chamber that had a fourth phase or exclusion zone, and we added turmeric in various concentrations. And we found that the concentration that would be typical inside your body expanded the exclusion zone water. You get more just from turmeric. And then we published that. And we looked at basil. We found the same thing. We also, in a subsequent paper, we studied ghee. And we not only found the same thing, but the exclusion zones grew enormously, maybe more than we'd seen in almost any other circumstance. So ghee, the concept from, I guess, dating from Ayurvedic times, that ghee is promotes health. it does. I believe it. And I think we figured out why it might do it, just like the other substances. So we also studied aspirin, which, although produced by pharmaceutical companies, it's natural. It comes from the bark of the willow tree. We found the same. We even tried Tylenol. I don't know if you have that in Australia, but it's kind of common aspirin substitute, and we found the same. There were one or two more I'm having trouble recalling. So anyway, going down the list of simple expedients to improve your health, that's the third one. The first was drinking water. The second was drinking easy water, and this is the third one. And the fourth one is sunshine. So where you live in Brisbane, I think there's plenty of sunshine. Where I live in Seattle, in the wintertime, sunshine is a rare commodity. There's lots of gray because we're right at the edge of the Pacific Ocean. And occasionally when the sun comes out, everybody feels good. So why do we feel good? Of course, the usual explanation is it's a purely psychological effect, which is not easy to dismiss. It might be. But there's another, that, as I mentioned earlier, that the sun is generating huge amounts of infrared energy. And you're absorbing that infrared energy. So you're absorbing it in pretty much all parts of your body. Your face is exposed. Your head might be exposed. And you're absorbing that infrared. And that infrared, once again, builds easy water. So it's possible that the reason you feel better is, at least in part, that in your brain, you're receiving lots of infrared energy, and that infrared energy is building easy water in the cells, in the nerve cells and other cells of your brain. And that's why you feel better. The fifth one, almost as an extension of that, is the sauna, or sauna, depending on what country you come from. And what is the sauna? Well, it's basically heat. So you walk in, whether it's damp, moist, or dry, it's still heat, pretty extreme heat. And heat is pretty much equivalent to infrared energy. It's not exactly the same, but, for our purposes, let's call it that. And you're sitting without clothing, or without much clothing, and you're absorbing this infrared energy from outside. And it's building easy water inside your body. So you get out, and when you get out, you feel better than when you walked in. I had that experience. I don't do this very often, but I was in Finland, and way up pretty far north in Finland, and I was at a conference, and I presented at the conference, and I was still jet lagged. It was a difficult day for me. And then in the evening, they said, we're having a party. So, okay, I go to the party. They load us in the buses and take us to this remote place where we had really good food. And I was just waiting for an announcement that the bus is about to go back to your hotel, load the bus, and we'll take you back home to your hotel so you can go to sleep. So the guy got up to the microphone, and instead of saying that, he said, okay, the sauna is now open, and we have 3 different kinds. And he was explaining, and I said, oh, shit, that's not what I want. I want to go back to bed and go to sleep. But, everybody was doing it, so I joined them, and I went into, I can't remember which one it was, sat there for 20 minutes, I had some conversations with the people, took my shower, went back to the party room, and I tell you, I felt like I just had 8 hours sleep. I was raring to go. I was ready for the next day. Just 20 minutes in that sauna. And I think the explanation could be really simple. It doesn't matter what's ailing you, whether it's a headache or a depression or muscle aches or whatever. You feel better when you come out because what this energy does is build easy water inside your body, and the easy water is necessary for good function, for proper function. So that's 5 of them. Let's see, what was the 6th one? Failing me. Pressure it's not the one I was thinking of. But it turns out we did experiments, and if you exert pressure on water, the ordinary liquid water turns into EZ water. And the reason we studied that was we were studying hyperbaric. We were considering hyperbaric oxygen therapy, and this kind of therapy. You may know, or you probably do, this is good for wound healing, but it's not just wound healing. The people who are dealing with it have identified something like 30 different syndromes that benefit from hyperbaric oxygen therapy. So again, the idea is, well, how's it working? Does the high pressure and high oxygen, do they have 30 different receptors inside your body, or is there one? And again, we're drawn to simplicity. Is it possible that the high pressure and high oxygen are responsible for building EZ water? And one of my students tackled this problem, and we published the result, and the evidence was positive. High pressure, well, by high, I mean just exceeding atmospheric. I don't mean super high pressure. A little bit of pressure does it, and a little bit of oxygen does it. And we can understand why that's so, because in terms of the pressure, the EZ water is denser than liquid water. And so if you squeeze the liquid water, it's easy to understand how it would convert to EZ water. And it's not only that, but oxygen, the EZ water actually has more oxygen per unit hydrogen than ordinary liquid water. So it's understandable for us why both of those expedients build EZ water. And so if you go back to the body, there are various practitioners who put light pressure on the body. I'm thinking of osteopaths, but there are other kinds of healers who will do this. And a possible explanation as to why it's as effective as it is by putting your hands there. You're doing a few things. You're first exerting pressure, which builds EZ water. And secondly, the infrared energy from your hands is being absorbed by the patient and in the region where the problem is. And so that can help reverse the problem. So this is there was another one that's escaping me at the moment, but maybe I'll think of it in short order. So those are a few of the relatively simple things that anybody could do to promote their own health.
They're all amazing. Amazing things that you're mentioning here.
Yeah, let me just tell you the other one that I'd forgotten. This is really important. I'm sorry, I don't like to interrupt you because I'm blabbering on and on.
Please.
But it's earthing or grounding. So I'm sure some of your listeners are aware of this, but it's if you take a walk on the beach barefoot near the water, you do it for 20, 30 minutes, and you feel really good afterward. And that you're electrically connected to the earth when you're barefoot. Or another example of doing this is in Japan, it's very common, the mud baths, high conductivity, you immerse yourself in the mud bath, and you feel better when you get out. It's also connected, you're connecting yourself to the earth. Or if you go walk into the ocean, you're, again, connecting yourself to the earth. And in all of those situations, most people report that they feel good afterward. And so, of course, the question is, well, why do you feel good? So, and I think the reason is that the earth, unlike what at least we're taught in the U.S., I studied, in my undergraduate courses, I studied electrical engineering. And no professor ever would even hint to me that the earth was anything other than neutral. If you plug the electrical cord into a receptacle, you're connected. The neutral prong, the third prong, is connecting to something that is neutral, right? And it shocked me to find out that was not the case at all. That the earth is actually negatively charged. it started with a Russian guy who was in my lab. And this Russian guy, just before he's returning, he was in my lab 6 months. And Andre was a clever and knowledgeable guy. And we had a discussion just as he was leaving for Russia that very afternoon or evening. And he's talking to me about the Earth's electrical field. And I said, Andre, what are you talking about? You mean magnetic field, right? He said, no, I mean electric field, completely distinct from the magnetic field. He said, don't you know that up in the ionosphere, there's positive charge and the Earth is negatively charged? And between the positive and negative, you have an electric field. And the magnitude is something like 100 volts per meter at the Earth's surface, which means if you're standing up, and that means you've got a 200, if you're 2 meters tall or something like that, you've got 200 volts between your nose and your toes. So, and I couldn't believe what Andre was talking about because it didn't make any sense. How is it possible that I never heard of this, that I studied electrical engineering, that I had deep interest in electricity, but never. So I went to bed skeptical. Next morning, one of my students comes in and brings the lectures of the famous Richard Feynman, the Nobel laureate, who people consider the Einstein of the second half of the previous century. And he had given lectures at Caltech, and they're put into 3 volumes. Someone did it, I think, after his death. And practically every physics graduate student studies those books here, at least in the US, because the guy was funny and he could explain things clearly. So he shows it to me. He opens to volume 2, chapter 9, and there was the whole chapter is on the electric field of the Earth and the negative charge of the Earth itself. So the Earth is not neutral, it's negative. When you connect yourself to the Earth, you're connecting yourself to a vast reservoir of negative charge. And so what happens? Well, if you have some cells that are not very negative, and you connect yourself electrically to this huge reservoir, the negative charge pours into your body. And as I mentioned earlier, it builds easy water. And so I think that's what's going on. It's why if you immerse yourself in any of those situations, walking near the water, or immersing yourself in salt water or mud baths or whatever, you're connecting yourself to this reservoir. And simple explanation is that the negative charge pours into your body, builds easy, and you feel better. And I think most people do feel better. I remember one incident when I was a kid, and I don't remember a whole lot from when I was a kid, but this incident really struck me. I must have been 10 years old. I was with my friends, and we were at the beach in Brooklyn, New York, where I grew up. And, in the summertime, you have to escape the heat. And so people go to the beaches, which are not so bad, but very crowded. And we were playing around on the beach. And toward the end of the day, hey, let's bury one another. So I volunteered, and they dug a big hole, and I was buried up to my neck. It was near the water. I was a bit worried about the tide coming in. But what I remember is I have rarely felt as wonderful as I did while buried. And it was getting time to go home. They wanted to unbury me, and I just didn't want to get unburied because the feeling, it almost beats sex. It's a wonderful, absolutely wonderful, wonderful feeling. And I remember it. And so there have been a lot of biophysical studies on trying to explain the positive effects of earthing or grounding yourself. There are various points of view, and the point of view that I would maintain is a good chance that the positive effect of grounding yourself comes very simply from the negative electrical charge that pours into your body, creates easy water, and promotes good health throughout your body. So that was the 6th one that I had forgotten about. And now we have a 7th one, which is pressure and oxygen. So
that's amazing.
Truly
amazing.
Yeah, it's interesting, and it's so easy, but the last one, we've disconnected ourselves So we have soles of our shoes that are non-conductors. And by the way, various companies are putting out shoes where there's a plug of metal so that you don't lose the connection to the earth. Another example of this, you're inspiring me to remember this, is my friend was telling me a story about that he read about George Washington, our first president.
Yeah.
After whom our university is named. And he, when he traveled from place to place, accompanying him was his physician, his personal physician. I guess the same thing happens in the White House now, where there's the equivalent of a hospital inside. So if our dear president is ill, he's got some good attention. So anyway, but George Washington brought his physician. One day, he said to his physician, I'm not feeling very well. And they were somewhere out in the forest. So physician said, just hug the tree. And so as the story goes, Washington hugged the tree for a few minutes. And after that, he said, I'm feeling fine now. So what the hell's going on? Why would hugging a tree make a difference? Well, the trees are basically full of negative charge. The cells are just like the cells in our body. And in fact, the electrical potential of those cells, plant cells in general, instead of being minus 60, minus 70, they're often twice that amount, minus 150 millivolts. So you're putting your arms around a whole lot of negative charge. And maybe that's the reason. You're just imparting negative charge into your body. So our body actually, bodies are not neutral. Our bodies are negatively charged. People don't think about that, but, we're 70% cells, and the cells are all negatively charged. I think because of this easy water. And even extracellular tissues like collagen and elastin and such also bear negative charge. They've also been demonstrated to be surrounded by easy water. So we're not neutral by any means, although, politically speaking, we might like to become more neutral, but we're negatively charged. And the more negative charge, at least the concept that I've come to appreciate, the more negative charge we have, the more easy water we have, the healthier we are. Okay, so let me stop that and wait for your next question.
No, it's really so good to hear everything that you're saying, because a lot of the things that you've mentioned, those 6 or 7 points, are things that we would usually get clients to do or patients to do as a part of their healing therapy. So these are things that we've known for a really long time that they have a beneficial effect, but I guess we've never understood the reason as to just why it works. And now you're providing evidence as to why these things are happening. It's
I think those are the explanations. As it was once said that even the most beautiful theory can be destroyed by one ugly fact. So one never knows. One can advance hypotheses, and they're just hypotheses that seem to fit, but one never knows in the future whether they're going to be proved wrong. I think not, but this is the direction we're going. And by the way, we just started this week. You didn't ask about. I seem to be in a talkative mood where we're actually looking at the role of healers. Now, I know this sort of thing. A healer on water, bound to be disbelieved or by so many, because it seems absurd that possibly that water could contain information of some sort, but we have a lot of good reason to think that that's the case, which I can tell you about if you ask me. But we're starting experiments with healers, putting their attention to water. So in the laboratory, we have a beaker of water. The healer comes in. We just ran the first sham experiment 2 days ago. Healer comes in, focuses his or her attention on the water in a positive way. And then we have 5 different standard instruments that we can apply to study the water to see if the physical chemical properties of the water have changed as a consequence of the healer. So we're really excited about that because there's evidence from so many quarters now, no matter how strange the concept might seem, that it actually works. So I organize each year the annual conference on the physics, chemistry, and biology of water. We had to skip the meeting that would have occurred right just now, October. We're going to do it next spring because of COVID. People obviously can't travel. I can't travel myself even. So running the show is not so easy. And each time we have a conference each year, 2 or 3 people will report the results of their experiments on water memory or water information. And I'm not talking about I'm talking about genuine, objective scientific experiments. There's also work by people who are spiritualists, like, for example, the late Masaru Emoto, and and most scientists will reject that if you don't know about the work. So he was a spiritualist, passed away 5 years ago. He would focus his attention on the water and think positive thoughts like, I love you, freeze the water, look at the crystals that formed, and they were beautiful. And then he would focus negative attention on the water, and the crystals were not beautiful. They were really ugly. And he's been criticized, and I think aptly so, because he would freeze 50 petri dishes and in one condition and 50 in another, and he'd pick out the ones that he thought best illustrated what he wanted to show. So it's called cherry picking, and a lot of scientists are dubious. However, I'm talking about experiments. By the way, the group that follows in his footsteps have now been beginning to adopt more scientific approaches to this, and it'll be interesting to see what comes out. But I'm talking about scientists experienced scientists who are now convinced that this is a real thing, including, by the way, Nobel laureate Luc Montagnier, who got his Nobel Prize for discovering HIV. He comes each year and will be coming to the next conference, which will be late April 2021. And his evidence is nothing short of amazing about water being able to store information and transmit that information. And so if that's true, and so far the evidence looks promising, if that's true, it means that information, subtle kinds of information, can be transmitted from one person to another, for example. So, you're standing next to a person and you feel something. You don't know exactly what it is, but you're getting some information. And that kind of subtle information, which nobody understands at all, seems to have an impact on nearby water. So his controversial experiment is, let me just tell you, because it's so interesting.
Please do, yes.
Yeah. So he's got 2 containers sitting next to one another, sealed containers. So there's no chance for any chemicals to be exchanged between one and the other. The first one has DNA, a short strand of DNA, sitting in water, actually diluted homeopathically. But basically, it's a short strand of DNA in water with a certain sequence. And sitting next to that is pure water, the kind of purest water you could imagine in a factory-sealed flask. And the 2 of them are sitting next to each other. And he supplies some kind of extraneous energy, 50 hertz, 60 hertz, something like that, just to power the system. And he says what's going on here is that the information, he hypothesizes, the information from the DNA or from the water surrounding the DNA is being transmitted to the nearby container of water. So how does he test this? He takes that water that's been informed, so to speak, and he uses the PCR system to build new DNA based, and the water that he uses is the water that's been informed, you see. So he takes the solids mixed with the water, and voila, you get lots of DNA out of it. and the sequence of the DNA matches the sequence of the original that was sitting next to the water. So, I mean,
incredible,
isn't it? It's totally incredible. He claims that two Italian groups have attempted to reproduce his results, and they've been successful in doing that. So, if this is true, I think it would be great if Luke can actually publish details. They've yet to be published. There are sort of sketchy descriptions, but if they're published and they're really confirmed, this is just simply amazing. It's not just him. I bring him up because he won a Nobel Prize, so he has some level of credibility because of that, and I give him a lot of credit because he's ventured into this domain. He knew from his late friend Jacques Benveniste, who was also espousing water memory and lost his career basically because he claimed to have evidence that water contains memory. So the bottom line is, it looks like it's real, even though it's hard to imagine. It's a little bit easier for me to imagine because I think of a memory of a computer, and the computer consists of silicon atoms that are organized in 3 dimensions, and each atom, in one way or another, can have one of 2 states. We say 0 or 1, but on, off, or whatever. And when you have a organized 3-dimensional array of that, with each atom occupying one of those 2 states, you have memory. Okay, it's the same thing that happens in your thumb drive. That's how it works. Now, if you think about the easy water, which I just told you about its structure, each sheet is full of oxygens and hydrogens arranged in a honeycomb fashion. The question is, could the hydrogen or the oxygen occupy 2 different states? You've got the order, which is the same as in the thumb drive, but do we have possibly two different states? Well, as far as I know, the hydrogen doesn't satisfy that, but the oxygen has actually not 2 oxidation states, but 5. You can find this in any chemistry textbook. It's common knowledge in chemistry. It's nothing radical or so. So if you think about the easy water, with each oxygen in that 3-dimensional hexagonal array having the capacity to occupy any or each of 5 different states, the capacity for information storage, it turns out, we calculated some time ago, it's more than 1 billion times the information density that now exists. Of course, nobody yet understands how the information gets in, how you can get it out, and this remains for the future. But I think this is the possible substrate, the easy water, for the storage of information. And now we can take the easy water and we can make a solid out of it at room temperature. Imagine a kind of water that's a solid at room temperature, just like ice, but room temperature. It was done by an Italian group led by Vittorio Elia, who's published a few papers on that. And we got so interested that we've begun doing it ourselves, and we could confirm that it works. So you have the possibility of having a solid kind of water that has the capacity for information storage. And who knows if in some years from now, your laptop computer will have water of some kind in solid water as the substrate for memory. So that excites me too.
It excites me as well. Just everything that you've been speaking about is just blowing my mind, Dr. Pollack.
Well, thank you. I appreciate it. Yeah.
You've given so much amazing, groundbreaking, thought-provoking information over nearly the last hour and a half. And I can't wait to go back through this podcast and listen to everything that you've said again and dig a little bit deeper into these different aspects of easy water and structured water.
Thank you for your kind comments. And I think I've gone through pretty quickly, and I do understand that sometimes when the speaker thinks that's perfectly clear, it's not necessarily perfectly clear. And many of the things I'm talking about are in that book called The Fourth Phase of Water. And it's become a really popular book. So I do recommend it. It's a book that's designed for non-experts, and it's got cartoons in it and a bit of humor, but it's a serious book that tries to describe the things I've been talking about plus more, a lot more. So yeah, it's been my pleasure. You're a great interviewer.
Oh no, look, I've got a great guest on. I think that's it's all really resonating with me, the information that you're providing. And Dr. Pollack, I'm so grateful for you to actually take the time to come on and speak with me about this topic. It's one that I'm really interested in, and I know that many clinicians and colleagues of mine and listeners of this podcast are also very interested. And what I would like to do is in the show notes, if you don't mind, is actually put up some links to your work and research and your books.
Sure, absolutely.
And maybe even also your laboratory, which is pollacklab.org. Is that correct?
I think it is, but if you email me, I can send you be sure that it's correct. I think that will get you there, but there's a more precise link, I think. So if you just email me, I'll get back to you on that.
Fantastic.
Sure.
No, I'm more than happy to share your books and links to your research and your website with our listeners.
Thank you.
Dr. Pollack, I'm just so grateful that you've taken the time. I understand you're very busy and you've got a lot of researchers and students underneath you who obviously require a lot of your time as well. So, it means a lot to me that you've given up that time to come and help spread the word about structured water. Thank you.
Well, I was very happy to do it because, for me, it's so exciting. So thank you very much, Daniel. Okay, and we'll keep in touch. Okay?
Absolutely. And Dr. Pollack, just in the time that we've spent together, I've got about 6 pages of notes and questions here.
Oh my goodness!
So if in the future you have time again, I would actually love to get you back on and see if we can answer some more of these other questions that I've now got on my mind.
Delighted. Okay. And I love to visit down under, but hasn't been possible lately. It's one of my favorite places.
Well, hopefully things change soon and you can make your way to Australia, and I'd love to catch up with you if you do come down here.
Okay. All right. Anyway, take care, and we'll be seeing you. Okay.
Great. Thank you so much, Dr. Gerald Pollack.
Thanks for tuning in. We hope you enjoyed the show. If you have any questions or comments, head on over to humanley.com/podcast and join the discussion. Make sure to follow us on Facebook and become part of our growing community of like-minded health professionals. Until next time.